And I'd like to start today by apologizing for my boring title here, especially as parasite population structure is fascinating as critical evidence of how diseases move through host communities. And understanding the population structure of a disease agent, and I'm going to use the term parasite throughout this talk, can help us predict how epidemics will spread throughout a community, and may also help us be able to prevent epidemics from starting if that's the goal. So understanding parasite population structure is particularly important if that parasite can infect multiple host types, which is the case for the zoonotic emerging infectious diseases that are becoming an international threat. So here I am plotting the predicted niche of Ebola virus from the range of the host of Ebola virus's host species. And within this range, there is virus strain structure with certain virus strains infecting certain host types. And it would be really useful to understand what leads to this spillover and breakdown of strain structure between host types. So I don't study the Ebola virus, but I study a maybe more tractable system, and I'm interested in these questions in a multi-host parasite system. So given a community of two Daphnia host types, we can see we have one Daphnia with a sort of rounded head here and one with a curved helmet. We can imagine two sort of extremes for how transmission might work if a parasite were to be introduced into this system. So in the first, we can imagine that a parasite, given parasite strain, would only be able to infect one host species, and that, so here we can see that a red parasite strain is infecting that red, or the round-headed Daphnia, and the purple parasite strain infecting the curvy-headed Daphnia, and that we can see the strain structure across two different lakes. So the red strains would be more closely related to each other than they would be to the purple strains. On the other hand, we could perhaps see this transmission with parasites being able to transmit within a lake, infecting all the different host species found in that lake. And in this case, we predict to find the parasites in that lake to be more closely related to each other, even if they're infecting different host species. And perhaps there's some sort of spectrum here between these transmission dynamics. So I'm really interested in looking in my system how much transmission is occurring within versus between host types. And in the future, I'm really interested in tackling the question what parasite traits, host traits, and host community structures can lead to these patterns. And I work in a great system to study this question. So I work in a series of lakes close to Ann Arbor, Michigan, and there are these darker blue ones highlighted here. They're all mesotrophic to oligotrophic lakes, home to between 1 and 5 Daphnia species. These Daphnia are key members of lake foodwebs, and they're also infected by a variety of parasites. So the parasite I'm going to talk to you about today is the well-studied bacterial parasite Pasteurella ramosa. It has been found to infect all of these host species here, but it's unknown. It's unknown how much transmission occurs between those different host species. So in a given lake, we could have a pool of parasites that can all infect the host species that are found in that lake, or we might have a pool of parasites that's composed of cryptic parasite species that each has their own host that it infects. So a little bit more about transmission of this parasite. So Daphnia graze for their algal food. As they're eating, they're going to come in contact with spores of the parasite. The parasite infects via the matching alleles model. So that means that a given host genotype must match the parasite genotype for that parasite to infect. But if it matches and the parasite infects, the parasite first sterilizes the host, and then it replicates in the host hemolymph. So you can easily see an infected Daphnia versus a healthy Daphnia here. When the Daphnia dies, 100,000 to a million spores will burst out of that carapace and into the water and restart this infection cycle. Annual epidemics are seeded by spore banks, so these spores can overwinter in the mud of lakes. So I'm going to start by showing you some field data from last year. I'm showing you dynamics in 5 different lakes here. And on the y-axis, we have the proportion of infected Daphnia throughout the epidemic. the different colors represent the different host species that get infected in these different lakes. And I want you all to notice that the size and the shape of these epidemics is really different in the different lakes. And also notice that the species that get infected is also different in the different lakes. Cedar Lake has an extremely large epidemic here. On the next slide, I'm going to just zoom in on the lower portion of this graph here so you can see what's going on in the other lakes. Again, it's an extremely virulent parasite, so even low infection rates can cause population level effects. Okay, so here's that, zooming in on that lower proportion infected range. I'm also showing you here densities of Daphnia hosts that were present in the lakes in August when these epidemics were getting started. So host density drives dynamics in many host parasite systems, but it doesn't seem to be doing that in this system here. So Little Appleton is a lake with about 3 times the host density as all the other lakes, yet it has a really tiny epidemic. Cedar Lake has a fairly low host density and it has a very large epidemic. Also notice that the size of the epidemic in each host species doesn't necessarily match the relative density of that host in the lake. And in some lakes, a given host species may not even experience an epidemic. So again, I'm trying to answer the question, how much transmission is going on between those hosts? Is transmission primarily local, or do we have different strains that are transmitting between the different host species? So in order to answer this question, I collected infected hosts and extracted parasite DNA. I used variable number tandem repeat primers developed by Ebert's group in Switzerland to amplify neutral alleles. And then these were analyzed for fragment length at the Michigan DNA sequencing core. So to give you a look at what that looks like, I got for each locus, I got data back for each strain showing the fragment length of a given allele. And then we can visualize this data with a principal components analysis. So here, each strain is a dot, and it's colored by the host from which the strain was taken. So we can see that parasites infecting Dentifera hosts, cluster over here, this is a fairly spread out cluster. Those affecting Retrocurva hosts are fairly differentiated, and those affecting Parvula hosts maybe overlap with both of the other hosts. This is interesting because on the Daphnia tree, Daphnia retrocurva and Daphnia parvula are sister species. So perhaps it's fairly easy for a parasite to be able to infect both of those host types. Daphnia dentifera are less closely related, so perhaps it's more difficult for those parasites to make that jump. And then going back to my PCA, I'm now coloring the strains by the lake from which these parasites were taken, and perhaps some more structures coming out here. So in that sort of larger dentifera cloud, which I showed you before, 2 lakes seem to cluster within that cloud, Cedar and Little Appleton. And in the Retrocurva and Parvula cloud, we have clusters for North and Middle Lake. This is easier to see with a dendrogram here. This dendrogram is generated by Jacquard distance between the different parasite strains. And the tips of the dendrogram are colored by the host species that the parasite infected. If we looked at this cluster here of dentifera strains, and then we color the tips of the dendrogram by the lake, we can see that within those dentifera strains, those found in different lakes are more closely related to each other than to the others. So this indicates that perhaps parasite population structure is first determined by the host type, and then similar strains circulate within a given lake. Going back to the dendrogram again, colored by host species, looking at this cluster here, where we have different host types clustering together. And then if we look there, those are all from the same lake, which is Mill Lake. And so perhaps this is evidence that spillover does in fact occur, although it may be quite rare. Mill Lake was a lake with a high diversity of host species, so perhaps that diversity is needed for spillover to occur, but we're looking into that. So in conclusion, I would say that transmission of this parasite occurs mostly among members of a single host species in a given lake, and spillover is quite rare. However, it is possible that spillover occurs. I'm really interested in quantifying that rate of parasite spillover, but I've yet been able to get a parasite from one host species to infect another host species in the lab. So again, this doesn't mean it's impossible, it just means it's not a common occurrence. And in the future, I'd really also like to look at how the time of year that the parasite strains are taken, how that affects the relationships, because one could imagine that after spillover, the parasites specialize on a given host type. And finally, I mentioned that there are a number of parasites that infect Daphnia in our system, and they have different transmission mechanisms. So I'm interested in seeing if those different transmission mechanisms might lead to different population structures. So I'd like to thank my advisor, Megan Duffy, and I'd especially like to thank an undergrad who worked with me on this project, Becca Village, and then my lab mates who helped me in the field collecting strains and data. And I'd be happy to take any questions.
Do you think there's any advantage for a bacteria strain to be in one host versus another, in that it can produce more spores when it finally kills the host?
Yeah, that's a really good point. These, the Daphnia are different sizes, and they do have different life histories too, so some live longer than others. The parasite doesn't actually shorten the lifespan of the host. Its main virulence is in castrating the host, and then it uses the energy from the host to produce a lot of spores. Definitely larger hosts and longer lived hosts will produce more spores. Dieter-Ebert's system is a Daphnia magna, which is, those Daphnia can be a couple millimeters big, and our Daphnia are a lot smaller, so yeah.
So it's spillover that difficult to make because of the matching up uniform?
Yeah, so that's really interesting. It's difficult to get a given parasite strain to even infect a different genotype of the same host species in lab. And so you have to find the right genotypes for that strain to infect. So that process is thought to be